Okay, good morning, everyone. Before we start the lecture, I wanted to congratulate everyone for getting through the first exam last week. Your TIs are busy grading the exams. I think several of them have probably already finished and have released your grades to you. If your recitation section, your TI hasn't yet finished grading, don't worry. I'm sure he or she will probably be done in the next day or so. And then you will be able to see your exam scores. In addition, you should also be able to see your current standing in the class in terms of your overall letter grade right now. For those of you who maybe are not where you want to be at this point grade-wise, don't worry. It's still early in the semester. There are plenty of opportunities to get more points because there are a lot more assignments, quizzes, homeworks, and we still have a couple more exams as well. If you feel like you really struggled with the first exam and you could use a little help, maybe a little guidance, don't be shy, right? I'm happy to talk with you and maybe discuss different types of study approaches or study strategies that you can try. So I encourage you, send me an email and you and I can, find a time and we can kind of talk about your situation. Obviously, it's better to do that now rather than wait, later, right? So let me know, and I'm happy to meet with you to talk about your situation. All right. I think now that we've finished the first exam, we're deep enough into this class that you are really getting a sense of what molecular cell biology is all about and how this particular kind of realm of knowledge that has to do with the living cell and how the living cell functions and all of the different types of biological molecules that define a living cell and therefore define life on Earth, right? That's what MCB is all about. I know, the majority of you, when you started at UH, you probably declared a biology major, either BA or BS, as your choice. Because here, of course, as a freshman, you have to declare a major. And biology makes sense because, hey, everybody took biology in high school. You knew what biology was about, right? And for those of you who wanted to or are thinking about moving on to things like graduate school to do more biomedical research, or even health professional school like medical school, nursing school, PA school, dentistry, optometry, all of those things, probably figure, yeah, a biology major, right, can get me there as well. So this is a good time, though, to really realize that we do have a strong molecular cell biology major as well, right? A Bachelor of Science in MCB, which is one of your major options. And what I'd like you to think about is, now that we have learned a lot about the molecular nature of these different molecules that make up living things, if this is a particular area that interests you more than necessarily the more macro sense of biology that might involve things like ecology, environmental sciences, things like that. Now, don't get me wrong, those studies are very important and valuable, right? But depending upon what your interests are, the MCB major may be something better suited to you, right? Not only to help you prepare for, let's say, medical school, the MCAT, but importantly, to put you in a better place so that when you're in medical school or PA school, for example, or graduate school doing biomedical research, that you are better prepared to succeed there. If you're interested about the MCB program, again, shoot me an email. I'm happy to talk about it with you. I am the faculty coordinator of the program. Maybe that's no surprise. That's why I teach Bio 275. You can reach out to me or talk to one of the NatSci advisors, right, who can help you figure out if the pathway you're on so far can easily take a little fork in the road and allow you to finish as an MCB major. Okay, because it's not too late to at least ask questions and find out about that option. Okay, so let's take a step back, right? We've been talking about the genome of living things as being the collection of DNA that's contained in the cells of that living thing that provide all of the instructions that code for things like the proteins, right, and other types of molecules that need to be managed and assembled and coordinated and interact to make something alive. Right? So I know all of you are familiar with this idea of DNA being the blueprint for life, right? And in this class, what we want to do is sort of look at it in a different way, right? Rather than just saying, yeah, it's the blueprint of life. What does that really mean, right? On the scale of molecules. So if we're talking about blueprints, I think, blueprints are something that we often think of when we think about architectural blueprints, right? So if you think of a master set of blueprints for a particular architectural project, like this house, for example, right? There's going to be many, many pages of blueprints, all part of the same binder. And the different pages contain different types of information. Right? These different types of information that are all part of this master blueprint, you can think about them as being specific for different types of contractors to know what their job is, what they're supposed to do, and how they are supposed to contribute to creating this overall brand new house. So from this one big common master blueprint, the different types of contractors and subcontractors follow the instructions that are relevant to them. So let's imagine that these 6 different subcontractors here working on the house. You have cement people, carpenters, masonry people, plumbers, electricians, and then the people who install things like fixtures like windows. Right? You can imagine each of them might have the same set of master blueprints at their disposal, right? But they only use certain sets of instructions within those blueprints to do their specific contractor job. Right? And together, they work together to be able to create something that is cohesive and the intended design, a very complex design, a fully functional house. So think about it this way. Our genome is that master blueprint. And every single one of your nucleated cells in your body carries a copy of that master blueprint in its nucleus. And that master blueprint, just like the big master blueprint that the contractors have access to, contain all the information that's needed. Right? Not only all the information to determine what the different types of cells are in your body, but for those individual different types of cells to follow specific subsets of instructions from that blueprint. In other words, only the subset of genes that are relevant to being a nerve cell, a muscle cell, hair follicle cell, a gland cell, etc. So I think one of the really fascinating things to think about is contained in this master blueprint that each one of these different types of cells in your body has in it. And by the way, I don't know if I had mentioned this before, but I don't know if you've ever considered how many distinct different types of cells actually make up the human body. Right? There are roughly 200, 220 or so different discrete types of cells in your body. Right? And that means each one of those carries the master blueprint in them, but only uses a certain set of genes, just like the plumber would carry the master blueprint around, but only pay attention to things like where the pipes are, or the electrician would pay attention to only the instructions that tell her where the wiring should be. Right? So the interesting thing is, not only do these sets of instructions determine that a nerve cell looks like a nerve cell and behaves like a nerve cell, or a muscle cell looks like a muscle cell and behaves like a muscle cell, right? Different sets of instructions got them there in the first place, right? Of course, because originally, if you go back in time, I mean, here we are sitting here today, right? Your body is made of somewhere on the upwards of 30 trillion different cells, right? 220 or so types, but 30 plus trillion of them are what you see when you look at yourself in the mirror. Right? Each one of those going back in time, of course, originated from that one single fertilized egg. Right? So that one single fertilized egg. So when we look at these wonderful images down here of different types of cells, just to give you a sense of the diversity of different shapes, the morphology, functions of the cells, right? Among them, right, is this egg right here, right? That is at this moment being visited by these little sperm. You can see that's what they are on the outside, and maybe one of them has already entered, fertilizing this egg, therefore completing, right, the bringing together of two haploid human genomes, one that came from mom, that was in the egg, one that came from dad, right, in the sperm, to now create a complete set of a diploid human genome, right, two sets of chromosomes, right? And that two sets of chromosomes, think about how amazing this is, right? That original set of DNA molecules was you, right, 20 plus years ago, right? And that original set of two human genome copies, right, began to replicate, divide, and eventually, decades later, a couple decades later, here you are, 30 trillion cells. So all of the instructions for making the 220 or so different types of cells in your body are contained in those genes. And importantly, once you have cells that have become nerve cells, muscle cells, gland cells, whatever, right, they follow only a subset of genes to perform their proper functions. Okay. So remember, every single one of your cells carries a complete copy of your unique genome given to you half from mom and half from dad. Different cell types in our bodies use different sets of genes in those master blueprints to define how they appear, where they reside, and how they function. So hopefully just thinking about it this way makes you realize that the control over what genes actually get used at any given time by a particular cell is a very complicated, highly orchestrated process, right? And this process is what we can call kind of generically gene regulation, right? How does a cell control what genes out of the, let's say, 20 to 25,000 genes in the human genome that are part of the master blueprint, right, do they actually use? And how do they control that? So let's talk about the importance of gene regulation. Right? Not as you see it today, when we talk about our fully developed adult bodies with our different types of fully differentiated cells in them, but let's start way back at the beginning of development. Right? And let's say, for simplicity's sake, we won't use necessarily a human embryo as an example. Let's use another animal model that's been very widely well studied for decades now, the fruit fly or Drosophila. Right? So here you see an early Drosophila embryo. Right? This is at the point when it looks nothing more than just a tiny little, I don't know, a little grain of rice or something like that, right? At this point, it's well beyond simply being a fertilized egg. It's already divided many, many times over. And so maybe this little grain of rice, this early embryo larva, right, consists of, let's say, maybe a few thousand cells at this point. Okay. What you can do is you can use certain types of antibodies that are specific to binding different types of proteins that you might be interested in. Right? What you say, why are we interested in proteins? Well, remember, proteins are the products that are coded for by the genes. Genes that, of course, are transcribed into RNA that then gets edited into mRNA, and then the mRNA gets translated into the polypeptide chain of amino acids that hopefully will fold up to form the proper three-dimensional protein. So researchers were wondering, what are the key early genes that get turned on in something like a Drosophila larva that start to determine, right, the growth pattern of the body as it forms? And so there were certain genes that we call early developmental genes or homeobox genes that are oftentimes referred to as Hox genes, right, that we now know are critical in early development. Very early on, these genes are expressed so that protein products are made from them. And these protein products then, right, control the fate of the particular cell that's making them, so that that cell will become something else later on as the embryo, the larva, develops. So here what they've done is they've stained this early Drosophila embryo with different antibodies that are specific for different early homeobox genes, right? Or I should say the proteins that are made by these homeobox genes. And each antibody has a different sort of little fluorescent molecule linked to it, so that if you then, shine a particular wavelength of light, you get these different colors, right? So if it were not for this type of antibody staining and the special light, all you'd see is, let's say, a couple thousand individual, maybe like white cells, right? You wouldn't know what's going on inside of them. But if you stain them this way, you suddenly see that at this point, early gene regulation steps are taking place, right? So for example, these cells here at the front stain very heavily for this particular homeobox protein, right? But it's really localized to only this group of cells at the front. These cells in the middle here, right, don't stain, let's say, necessarily for any of these markers that are being looked for, but it's clearly defined. Once you get into this middle part, you can see alternating bands of cells that are expressing a certain homeobox gene that glows in green, whereas their neighboring cells are expressing a particular gene and protein product that stains in pink, right? So these early steps are decisions in a way, right? The cells are making decisions by switching on certain genes and expressing them and leaving other genes switched off, right? And those early decisions will ultimately go to form the body plan layout of the animal, right? So if you look at the adult Drosophila here, it's gone from one cell to a few thousand that we saw up here to ultimately millions upon millions of fully differentiated cells that make up the structures of the cell's body. But early on, you can see things like the banding pattern here that define the segments of the abdomen of the fly, right? Or maybe this dark region would be the cells that ultimately form the thorax. And then this part here, the blue and orange, form the head, right? So if you look at that map, it looks something like this. Drosophila researchers now know that the switching on of these particular early homeobox genes to make their protein products help drive downstream or subsequent gene regulation events in the cell. So that makes you say, well, if these homeobox proteins are these super important ones that get turned on in different cells, depending upon if those cells are going to become part of the abdomen or thorax or even wing or the jaw, right? What do these proteins do? These proteins themselves are transcription factors. Remember, transcription factors are proteins that bind to other regulatory sites in chromosomes and control other genes being turned on. For example, transcription factors bind to promoters of genes so that RNA polymerase can be recruited there and begin transcribing. So these early developmental homeobox proteins are critical decisions that are being made, because you can imagine, for example, let's just say this particular homeobox gene here, that let's just arbitrarily call it purple, right? If that's expressed in cells, then genes that are responsive to this purple transcription factor would be the next wave of genes that get turned on, and so on and so forth, so that eventually cells that expressed this early purple homeobox protein, right, go on to form this part of the fly's body, including the wings. So you look at this and you're like, yeah, that's pretty cool, and what they've done here is they've just sort of reminded you that a fly already is pretty advanced in terms of evolution, right? A fly is like well beyond simple individual cellular prokaryotes, right? Now we're talking about complex organisms that are invertebrates, that are absolutely eukaryotics. But early in multicellular animal development, right, these homeobox genes would have been kind of generic, right? That's what these sort of yellow or beige ancestral genes are meant to represent, right? So through evolution in the fly, they became a bit more specialized, right? So you can form the fly body plan like this. But okay, but that's a fly, right? Even though fly is pretty amazing and complex as an organism, you say, well, that's nothing compared to like a human being, right? Our body plan, must be far more advanced than this, and we probably abandoned this type of early homeobox gene expression a long time ago, right? No, but not so fast, right? Because if you look at the way we develop the same way, right? Early in the development of the human embryo, we have the human versions of these same homeobox genes that, granted, yes, it's a bit more complex than what's in the fly, but in this color coding little table here, you can see it's undeniably the same thing, right? And these all come from, again, can be traced back to the same ancestral homeobox genes way back in evolutionary history, and the expression of these genes to make these particular homeobox proteins early in human field development is what's going to lay out our body plan, right? So when you look at this figure, hopefully this really gets you thinking about the fact that, again, right, one of the themes we talk about in this class is that all life on Earth, right, is tied together, right, through this chain of evolution, right, that ultimately goes back to the use of DNA. Right? DNA that billions of years ago, right, started the process of evolution, instructions in the DNA that allowed for the production of different proteins that could do certain things within a cell. Right? If those things were good and useful, they were kept, right? Even as evolutionary branches of life began to diverge. Right? So to this day, we still use the same type of genes from our human genome that a fruit fly does from the fly genome. Right. So let's talk about then comparing different genomes of different organisms. So the human genome, our genome, is about 3.2 billion base pairs of DNA spread out over our 22 autosomes. And then keep in mind that this is just showing 1 out of every 2 that you have, right? Because let's just pretend that this is the set of chromosomes that you got from mom, right? Chromosome 1 through 22. And then you would have another set that you got from dad, right? That we're not showing here, right? And then you got the Y chromosome from dad, right? So the sex chromosomes are showed here, both of them. Right? And so within these 46 individual DNA molecules, right, that make up your genome, and that's what each of your nucleated cell has, right? These molecules contain about 20,000 to maybe 25,000 different genes on them, which are nothing more, as we've learned, than particular regions of the DNA that represent some sort of message that's going to be used. Maybe it's going to be converted into RNA and ultimately into some kind of a protein product. If we look at the fly's genome seems much more humble, right? It's only 122 million base pairs. That's far less than 3.2 billion base pairs in a person. But surprisingly, a fly still has about 15,500 genes in it, right? So here is the fly genome. It has 4 large chromosomes. And again, these would be pairs. So there'd be 2L, 2R, 1 from the mom fly and 1 from the dad fly, right? But it's quite a bit smaller than the human genome. And yet, the number of genes it contains on it, right, isn't proportionately that much smaller. So if we look at this here, we have about a 30 times larger physical genome in terms of number of base pairs, A's, T's, C's, and G's, compared to a fly. And yet we only have something like 1.3 times more genes than a fly does. So that's kind of, I don't know. I mean, what do you guys think of that? Right? Because we like to think of ourselves as so much more advanced than something like a little fly, right? A little fly that you would just kind of swat, right? If it gets into your apartment. right? But yet, you know, a fly only has something like maybe 5 to 7,000 genes fewer than a human being does. Right? So what does this tell you about gene regulation? What it should let you realize is that our enormous physical size of our genome, much of that, most of that, does not actually code for anything. So much of our genome is not coding, but at the same time, it's not necessarily junk. Right? It's not like filler material. I mean, yeah, some of it is, right? But most of it actually has a purpose. Right? And that purpose is regulatory. What do I mean by that? Right? We've been talking about gene regulation. So what I'm saying is most of the stretches of DNA that you have in your genome don't themselves code for a product. They are in charge of coordinating the expression of other regions. And those other regions are the genes that code for something. Right? And you say, well, that's kind of weird because if it's all just a string of DNA, how can one part of the DNA somehow function to control the other part? Right? You're absolutely right. Because it is just one long string of DNA that makes a chromosome. But what have we been learning? Right? The DNA in our cells is not just naked DNA. Right? It's always associated with different types of proteins that are bound to it as part of organizing it and as part of controlling, right, what parts of the genetic sequence are accessible by other proteins. Right? So these regulatory sites of DNA must be sites where many different proteins are binding, and those sites coordinate those proteins coming together so that they can ultimately influence the expression of a gene target. Okay. So if that sounds a little like kind of heady, it's kind of hard to grasp, let's start a bit simpler, right, by using a model of an organism genome that's kind of easier to grasp. So let's start with something kind of ancient, like a bacteria, prokaryote. So not surprisingly, the genomes of bacteria, things like E. coli, are a very convenient model to understand how genes get regulated in a living thing. So the E. coli's genome is really kind of meager compared to your genome or my genome, right? E. coli's genome is only 5.4 million base pairs. And it's all on one chromosome, one circular chromosome, right? Of 500 or so million base pairs. And that contains about 5,400 individual genes on it, right? Individual stretches of that one circular piece of DNA that actually represent the code for different gene products. Okay. I mean, in addition to the main circular chromosome of an E. coli, individual E. coli might also have other smaller circular pieces of genetic information floating around in them. Those are what we call plasmids, right? But the actual genome of the E. coli itself is uniform from individual bacteria to individual bacteria, individual E. coli. This is a map that shows the entire genome of the E. coli. And it's a bunch of concentric circles, each circle containing different sort of information about the functions that are being performed or carried out by the DNA. So right, the inner circle itself that goes from blue around to purple, this is the DNA itself that's represented here. And each one of the little banded regions, I think, is meant to represent a different gene that's part of this E. coli here. Right. So as you go outwards, these are different sets of data. We're not going to get into what these sets of data are because this is something that you would study if you were getting into more advanced molecular cell biology. You'd become more familiar with how to kind of read these types of maps, right? But suffice it to say that these peaks and valleys of these different rings are measuring something. For example, how much RNA, for example, is coming off of a particular region of the genome. And then as you go to the outside, this might be, you know, translated products that are coming ultimately from the original DNA down here, right? These translated products are then converted into different proteins. And the outermost ring here is a subset of maybe very important proteins that are made from the 5,000 or so genes that are contained here. All right. So this is kind of neat, right? Because if you think about this one individual E. coli individual, like this guy up here in the corner, what you're looking at is the entire set of instructions for this bacteria to be able to carry out all of its life functions that include things like instructions on how to make proteins that enable it to have a metabolism, to be able to process nutrients, to get rid of wastes, to be able to respond to different environmental changes. That's kind of neat when you think about it, right? That when you look at this map, you realize, right, all of that information is right in front of you in one spot here. So these types of simple examples of genomes are a good model for us to begin our discussion of how genes get regulated in response to different types of situations. Metabolism is one of the fundamental kind of living properties of all life, right? Metabolism is a combination of how you take in nutrients, for example, from the outside of the cell, and how the cell can take individual building blocks and create things like macromolecules from them. And at the same time, the metabolism of the cell involves taking complex big molecules and breaking them down again, right? So this constant cycle of building things up, breaking things down over and over again, that together is metabolism. So those two halves, the building and the breaking down, right? That is the anabolic processes and the catabolic processes. Let's start with the catabolic processes. Catabolism is the breaking down of molecules for raw materials. If we're going to talk about bacteria, let's talk about the lactose pathway. So lactose, remember, is a disaccharide. It's one galactose sugar bound to one glucose sugar. Right, so it's disaccharide. All right, so lactose is the type of sugar that you see in milk. Right? So the cell wants to get the glucose out of this lactose disaccharide. Right? So how does it do it? So in bacteria, you can imagine if you have an individual bacteria that suddenly finds itself surrounded by lactose sugars, hey, right, this is great. The bacteria wants to make use of this lactose sugar and break it down into glucose. So that involves three key proteins here. This first one here is this galactoside permease. Okay? This second one here is what we call a transacetylase. And the third one here is a beta-galactosidase. Right? Now, I do want you to be familiar with these particular terms as an example. You might say, well, why? Because when am I ever going to care about bacteria breaking down lactose? No, the point of this is, right, if you can kind of understand this pathway and the principles that we're going to talk about, this is applicable, right, to further discussions about how our cells and our body control certain pathways as well. Right? So dedicate yourself to learning this pathway as a model, right, to help you understand more complex pathways. So this protein here, the galactoside permease, is in charge of taking the galactose or the lactose that's floating around outside of the cell. So this orange thing is the membrane of the bacteria, right? So this protein is in charge of getting the lactose inside of the cell, where it's here. This transacetylase is in charge of adding an acetyl group. So here's an acetyl group, adds it to this lactose in preparation for other proteins to be able to then recognize the resulting shape. And the critical protein that does that is this galactosidase, right? It recognizes the acetylated shape of the lactose, and it's just the right shape then to break the bond, right? This glycosidic bond between the two monomers through that process of hydrolysis, right? It adds a water back in there to reverse the condensation reaction that put these together in the first place. And then you then liberate or free the galactose from the glucose, and the cell then wants to use the glucose for energy. And we'll learn about that later when we talk about things like aerobic respiration and things like glycolysis. right? So critically, when you look at this, you see why it's called a pathway, right? There are different steps along the way to get this lactose in, to kind of modify it so it's ready to be used, right? And then to actually have an enzyme, a protein that breaks the lactose. So these three critical proteins that are part of this pathway, they can only work to create the glucose from the lactose if all three of these proteins are available, right, to work together. So the production of these pathway proteins must be coordinated by the bacteria. It's no use if the bacteria makes a ton of protein number 1, but forgets to make protein number 2, right? And likewise, it could have buckets of protein number 3, right? But if it doesn't have any protein number 1, this pathway doesn't go anywhere. So in bacteria, in simple prokaryotic genomes, there's a great example of how different genes that code for different protein components of pathways can have their expression coordinated together, right? And in bacteria, they are often found together physically next to each other on the same stretch of that one single chromosome that makes up the bacteria, right? And this one stretch is what we call an operon, right? So an operon is a term that's mainly used when you're dealing with bacteria. We don't really talk about the way our genes are regulated in our body as having operons, because as we will learn, the way we do it is a bit more complicated, but it still builds on these principles. You can see here, this is a stretch in blue of that E. coli's genome, and you're zoomed in right at that part on that big map of the genome where you have the 3 genes that code for those 3 lactose pathway proteins, right? Here's the galactoside permease, here's the transacetylase, and here's the beta-galactosidase, right? You'll see that the 3 genes are right next to each other on the same stretch of DNA. And hopefully what you're thinking is, oh, I get it, right? Because they're together, they can be controlled together so that when the cell needs them, they're made at the same time in the same amounts, right? So together they form what we call the lac operon, or the lactose metabolism operon. So these 3 genes have a promoter region. So this is the region where RNA polymerase is going to come and transcribe this gene, making ultimately an mRNA that can then be edited to get the 3 separate mRNAs out of it that can be separately then translated by the ribosome to make the 3 different protein products, right? So this operon has this critical feature here. You see this sort of beige area labeled with O is the operator, right? This is basically the on and off switch that controls whether or not these genes get transcribed. Okay? Upstream of it, so you can see up here, there's another gene called the LAC-I gene, right? And that I stands for inhibitor. Okay? So how does this work? Normally, a bacteria like this E. coli wouldn't necessarily be sitting in a drop of milk, right? I mean, sure, it's nice if it finds itself in a drop of milk, right, where there's tons of lactose, but maybe normally, right, there are other types of sugars around it, and lactose isn't really one of those sugars. If that's the case, then why would it waste resources making these different lactose metabolism pathway proteins if there's actually no lactose for it to even worry about? It could be instead making proteins, using its resources to make proteins from other genes that are needed, right? Maybe to utilize a different type of sugar that's available. So because of that, right, since the catabolism or the breakdown of lactose all kind of depends on, well, whether or not you have any lactose to start with, right? It would make sense then that in the absence of any lactose, the lac operon should be shut down, right? So that this bacteria doesn't waste its time making these proteins that aren't needed. Right? So that's how this works. It's going to be a coordinated on and off expression of these pathway components, normally in a turned off state if there's no lactose. Right? So this is how that works. That LAC-I gene that is right upstream of these different LAC-Z, LAC-Y, LAC-A genes that make those 3 different proteins that are part of the pathway, This LAC-I gene is what we say constitutively expressed. That means at any given time, the bacteria is transcribing this LAC-I gene so that you then have a LAC-I mRNA that gets translated into this repressor protein, right? And this repressor protein, the R, is the right shape to then float around and bind to the operator part here. And when this repressor protein is bound there, it physically blocks RNA polymerase from being able to be recruited there, so no transcription of this region happens. This illustration at the bottom is really neat, right? Because this is actually the real three-dimensional crystal structure of this repressor protein on the operator DNA. So this yellow region of DNA is the operator DNA that's up here. And you can see it's bound up, kind of crunched together, because this big protein that's made up of two separate peptide chains, a blue peptide chain, a red peptide chain, together forms this repressor protein. You can see it is the right shape, right? This protein is the right shape to grab the operator DNA and kind of scrunch it into this shape that's not compatible for RNA polymerase to come in and sit there. Part of this R repressor protein has a binding domain for lactose. Right? So what is going to happen then is if lactose is absent, nothing will be bound in here, and this repressor protein can hold on to the DNA at the operator, keeping the operon shut down. But if lactose binds in here, what does that mean? That means suddenly there's lactose available, right? Lactose is kind of coming into the bacteria, right? And now the bacteria wants to use the lactose, make use of it, start breaking it down to glucose. The lactose itself is the signal that binds to the repressor protein. And in a moment from now, right, if we were to see what happens next, we would see the protein would change its shape and release the operator. Okay, so the repressor protein normally keeps the operator turned off, but in the presence of lactose, so lactose is now coming into the cell, right? That lactose binds to the repressor protein and changes the repressor protein's shape. And the repressor protein lets go of the operon, no longer blocking RNA pol2, right? So the RNA polymerase comes in and begins to transcribe, right? And then you suddenly start making then the mRNA that's going to get translated into those three lactose metabolism proteins. So now the bacteria can start using the lactose that it's found, right? Because that bacteria landed in a drop of milk, let's say, right? Hey, whoa, this is great, right? It's not going to waste this opportunity. It wants to turn on these proteins that help it now make use of the lactose. All right. Here's another example of a molecule that helps out with this process. There's a very important molecule, signaling molecule, known as cyclic AMP. So what is cyclic AMP? Here's the structure right here. It's cyclic adenosine monophosphate, right? And you look at this, what do we see here? We have an adenine base, a ribose sugar, right? And a phosphate group, right? And you're like, wait a minute. Those are the three parts that make a nucleotide. This is a nucleotide, right? It is a nucleotide, but it looks different. Why does it look different? Because this phosphate group hanging off of it at the 5' position, right? Normally, if you were chaining together a bunch of nucleotides together, what would this phosphate group be doing? It would be in charge of connecting the C5 over here with the C3 on the neighboring nucleotide ring. Right? But instead, look what's happened. That phosphate group is actually bound to this nucleotide's own C3 ring, right? Forming this funny little loop right here. So obviously, this molecule is not going to be usable as part of building a long-chained polymer, a nucleic acid, but right, as cyclic AMP, because of this little cycle here, it's a very important signaling molecule used in the cell. Again, that theme of once Mother Nature came upon certain molecules that are useful for something, like building nucleic acids, for example, right? If Mother Nature found other purposes for these same molecules, because now the shape of this is going to be slightly different, ever so slightly different, because the phosphate group is basically bound to itself, right? It's then recognized by other molecules in the cell. There's a particular protein, this light blue thing here, called catabolite activator protein. And you say catabolite. Oh, right, we're talking about the catabolism of something like lactose, right? So this is a protein that assists or helps in getting things like the lac operator active and turned on. So it binds to cyclic AMP, right? So that little dot over here, this dot is the cyclic AMP, binds to this protein, and that protein also binds as a dimer. So two of these cap proteins come together, each bound to cyclic AMP, and they form the right shape to sit on the DNA and help put the DNA in a good position for RNA polymerase to come. So here you see that outlined on the right, right? So the cyclic AMP cap complex will bind to the cap site, right? That's close to the operator, and that will assist in the RNA polymerase having an easier time finding the right fit or the right shape to sit there, right? So that encourages the RNA polymerase to start transcribing the lac operon. So here's a little tidbit that I don't want this to get lost because this is very important conceptually. If you have a lot of glucose present, right? Let's say the little bacteria that was in, the little drop of milk, right? Now suddenly, right, there's a ton of glucose, just free floating glucose in the milk, right? The cell would much rather just focus on using the glucose as is and not have to bother with taking something like the lactose, breaking it down just to get the glucose out of it, right? Because if you already have a ton of free floating glucose, why are you bothering to break down the lactose to get the glucose out of it? So, right, we have what's known as a feedback loop, because if you have a ton of glucose present, glucose itself actually inhibits the formation of cyclic AMP in the first place. And by doing so, the presence of a lot of glucose kind of dampens or turns off the lac operon, right? Because that's not really needed as much if you already have a ton of glucose floating around. So here then is an example of how things like the repressor protein and the cap protein are working, right? So normally you have the repressor protein that's bound to the operator region here, causing the operator region to be kind of bunched up so that RNA Pol2 can't come in and sit on it, right? Already you see the cap protein is there because the cap protein's in there so that when the repressor protein is released, for example, when lactose comes along and binds to the repressor protein and the repressor protein falls off, this cap protein can already help, right? Reshaping the DNA to make it better for the RNA Pol2 to come down and sit there. All right, so last slide. Those previous slides were talking about a catabolic pathway, right? The breakdown of, let's say, a nutrient like lactose, if it's available, in order to break it down and get something useful like the glucose out of it, right? And in those situations, those types of operons or collections of genes that are meant to work together in the bacteria, right, are normally kept shut off because most of the time there is no lactose to even worry about. And when the lactose appears, right, the lactose turns on the lac operon so that you then make the proteins that can then go to work doing something to the lactose. But what about, instead of catabolic pathways, what about anabolic pathways, right? The pathways in the cell that are involved in always building things up rather than tearing things down. So anabolism is the creation of new molecules starting from raw materials. Many of these new molecules that are being made are molecules that are just needed all of the time. For example, amino acids. Certain amino acids are made by the bacteria itself, like tryptophan. Right? So tryptophan, here's tryptophan down here, right? Remember, it's an amino acid. Here's the little R group of the tryptophan. There's not a time when the bacteria doesn't need to have tryptophan, right? Because how is it going to make any proteins if it's missing one of the 20 amino acids? So the cell is always doing things like making tryptophan. Right? So the way that works is similar. You would have several genes that are related to different proteins involved in making tryptophan. They're under control of an operator that's going to influence whether RNA polymerase gets recruited. Right? And in this case, normally, this repressor protein, okay, is the wrong shape to bind there to the operon. So if it's the wrong shape to bind there, then RNA Pol II is always able to transcribe this operon, and the cell is always able to have the protein components it needs to make tryptophan. Right? If, however, you have way too much tryptophan, right, then maybe what the cell says is, whoa, 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 whoa. Yeah, we need tryptophan, but we have way more than we need right now. Maybe we'd rather use our resources to kind of make some of the other amino acids to make sure that we have enough of those other things, right? So this is a different type of feedback loop. Here, if you have too much tryptophan, the tryptophan binds to the repressor, and this time puts the repressor protein in the right shape to bind to the operon, right? So in other words, in response to excessive tryptophan inside the bacteria, that will deactivate the trip operon, right? So that the bacteria no longer wastes time making more tryptophan. Okay. And that's what's shown here, because here's the trip repressor protein. Right? When it's bind to tryptophan, that's what these little amino acids are in here. It is the right shape to buckle in to the DNA at the operator, thereby blocking the RNA polymerase from transcribing the trip operon, therefore shutting it off. Okay. All right, so that's where we'll stop today.